Hello everybody. I wanted to take a moment and explain to you transcription and translation in a different way. I find that my students often have a hard time imagining it or relating it to something. And so I came up with this chef analogy actually last year, and it seemed to work out pretty well. So I'm going to walk you through transcription and translation using a kitchen as kind of the model for this. Transcription is actually the process where we take normal DNA and we turn it into RNA. And for our purposes here, we're going to say that it turns it into messenger RNA, but there are also two other types that we're going to deal with called ribosomal RNA and transfer RNA. And so our goal today, the end goal of transcription, is to make a single strand of messenger RNA from our DNA. All right, and so if we liken this analogy here, we have our DNA here. If this were a kitchen, the DNA would be in the nucleus, so in a cupboard, and the DNA would actually be a cookbook, and that cookbook is full of recipes. Each one of those recipes is a special recipe to make a protein, but the body cannot make that protein until it pulls the recipe out of this DNA cookbook. And so the main guy that does this is this big purple thing that you see right here. This is your strand of DNA or your cookbook. In this case, this guy right here is called RNA polymerase, but he is our head chef. And whenever the body needs a recipe, the head chef comes and he goes to the specific place on the DNA that makes that protein, and he reads it and he creates a separate recipe. This red line that you see coming out here is messenger RNA, and so you have your DNA, and this polymerase, this chef, makes a messenger RNA strand from the DNA. And so when he's done, you end up with a half a strand of messenger RNA here. You have a double helix here, you have a single one here. Now, it's interesting to note that this chef does not always translate things properly. In fact, he realizes that in order for this strand of messenger RNA to be stable, he can't use the same letters here as he does here. And so any place in a messenger RNA strand that should have a T, a thymine, it is instead replaced with a uracil, a U. And so the chef is actually adding his own special goodness to this messenger RNA recipe. And that is basically transcription. You have DNA, you have RNA polymerase that rides along the DNA, and he makes a separate copy that breaks off and becomes a messenger RNA strand. There are some terms that I didn't discuss here, like elongation and exons and introns and things like that, but this is just for basics to understand transcription. All right. Translation is actually the process of taking that messenger RNA strand and turning it into a protein. Well, not turning it, but making a protein from it. And so translation actually uses those other two RNAs that we talked about. It uses ribosomal RNA and transfer RNA to create the protein that we need. All right, and so there's also some other words here called anticodons and messenger RNA, and I'll use them in my analogy here as well. And so we had the nucleus, which held our DNA, and from it, our messenger RNA strand was made. When that messenger RNA strand is made, it actually leaves the nucleus. And so if this is your nucleus right here, you can see that there are pores in this nucleus. Well, the pores allow that messenger RNA strand to land on the rough endoplasmic reticulum, which you can see is holding ribosomes. That rough endoplasmic reticulum is your actual kitchen there. It's where the protein will be made. And these ribosomes, I've highlighted them here. This pink blob is ribosomal RNA. and he is what we call our sous chef. So if there's a head chef who decides what's going to be made, the sous chefs are the ones that actually do all the work. And so this ribosomal RNA is our sous chef. You have this messenger RNA strand, which is our recipe, and our sous chef is reading this recipe and telling the transfer RNA, which will be our line chefs, they bring the ingredients. So this transfer RNA, do you see how it's shaped like a T? These guys are your chefs, your line chefs. They go out, they find the ingredients, and they bring them when the ribosomal RNA or the sous chef tells you to bring it. And they drop off their ingredient, and then they go out to go find more ingredients which are found in the cytoplasm of the cell. Now these ingredients are called amino acids. If you remember, we said a protein is made of amino acids. So the whole process and the whole purpose of DNA is to create proteins. It is nothing but recipes for the proteins that make you who you are. And so in this case, this poor body, he needs a protein. He needs that protein really quickly. And so DNA is made in the nucleus and it's sent out to the rough endoplasmic reticulum where it has these ribosomes on it. And if you highlighted in onto that ribosome, you would see this happening right here. And it will grow and grow and grow with amino acids until the protein is finished and it's all good. Now you need to understand that there is something called an anticodon. When this ribosomal reads this messenger RNA strand, he reads it in groups of 3. This is called a codon. That is the actual recipe. Well, the transfer RNA guys, they have to read that recipe upside down. And so the recipe that they read upside down is the exact opposite of the codon strand. So here's a codon, and on the transfer RNA is your anticodon. I think that's pretty much it. I have put here for you all the parts. So we go back, and that head chef was our RNA polymerase. The recipe book was our DNA, and the specific recipe is our messenger RNA. That messenger RNA leaves the nucleus and goes to the kitchen, which is the rough endoplasmic reticulum, where a sous chef, which is your ribosomal RNA, binds onto that recipe and tells the transfer RNA what to bring. Those specific directions are 3 at a time, and they're called codons. The line chefs, which are those transfer RNA, will use their anticodon to read the recipe upside down and drop off their ingredient, which is an amino acid. And finally, the finished product, when the messenger RNA strand is fully read, you end up with a protein. A protein is a long chain of amino acids or a polypeptide. That's all. If you have any questions, you feel free to contact me. Thanks.